is extremely abnormal considering T-cell progenitors are primarily found in the thymus. So I just introduced a couple of situations where abnormal cell production, or when cell production goes awry, blood cell production goes awry. So now I'd like to give you a brief overview of what happens during normal blood cell production, or hematopoiesis. So hematopoiesis occurs throughout the lifespan of an animal, and it begins when long-term hematopoietic stem cells gradually lose their self-renewal capacity and commit to either the lymphoid lineage or the myeloid lineage. The common lymphoid progenitor, or CLP, can give rise to all classes of mature lymphoid cells, such as B cells, T cells, and T cells, and certain subsets of dendritic cells. Similarly, the common myeloid progenitor, or CMP, can give rise to certain classes of dendritic cells, granulocytes, macrophages, platelets, and red blood cells. Under physiological conditions, CLPs do not give rise to mature myeloid cells and vice versa. How this population of pluripotent and multipotent progenitor populations commit to one of the lineages is very hotly debated, and there's a variety of different factors that are postulated to be important for this particular decision-making process. One of those factors is cytokines. So cytokine stimulation of cells can result in a variety of different cellular functions, including promotion of cell survival, proliferation, and differentiation and activation. Cytokine signaling is relayed into the cell via a variety of different signal transduction pathways, one of which is the mitogen activated protein kinase pathway. So the MAP kinase pathway is activated by cytokines as well as growth factors, serum, and osmotic stress. Ligation of the cytokine receptor with its cognate ligand triggers activation of a variety of different molecules, including RAS, which is a membrane proximal protein, which can then, when it's activated, can phosphorylate and activate RAF, which can then phosphorylate and activate MEK, and then phosphorylate and activate ERK. Activated ERK can affect transcription of a variety of genes that are important in cell growth, proliferation, differentiation, and survival. So some particular cytokines that are able to activate the MAP kinase pathway include GM-CSF, G-CSF, and IL-3. And all of these cytokines are important for myeloid cell development. So while it's known that these cytokines can activate this pathway, it's unclear exactly what role or how necessary this pathway is in myeloid cell development. So Jalen Su, a previous graduate student in our lab, wanted to address this question, and she did this by taking hematopoietic stem cells and culturing them in methylcellulose culture, either in the presence or absence of MEK inhibitors. And as you can see, in the presence of MEK inhibitors, there's a significant decrease in the plating efficiency of these cells. Also, looking more in an in vivo situation, she took siRNA against MEK1 to knock down MEK1 expression in HSCs and injected them into recipient mice, and then analyzed peripheral blood for the presence of MEK1 positive myeloid cells. And as you can see, when MEK1 is knocked down, the percentage of MEK1 cells is decreased. So kind of taken together, this data seems to suggest that the MEK-ERK pathway indeed can play, or has a role in myeloid cell development, especially for HSCs. So, as I said, RAS is a membrane proximal protein, and it is able to activate the MAP kinase pathway, but additionally, it can activate a couple of other pathways as well, one of which is the PI3 kinase AKT pathway, which this pathway can cause expression of genes that are primarily involved in cell survival as well as cell growth. Additional pathways that can activate are the RAL-GDS pathway and the PLC epsilon pathway. So RAS is of particular interest to researchers because it's been found to be mutated in approximately 30% of all cancers. With regards to leukemogenesis, or with regards to leukemia, it's been found to be mutated in approximately 10% of AMLs, 40% of JMMLs, and 30% CMMLs. However, because RAS is a membrane proximal protein, it's able to activate a variety of different signal transduction pathways, so the systematic deduction of which pathway is involved in oncogenic RAS signaling is necessary. So one of the pathways that RAS can activate is the PI3 kinase AKT pathway. So I should also mention that animal models featuring oncogenic RAS have been shown to predominantly give rise to MPN JMMLs with 100% mutations. As I mentioned, the PI3 kinase AKT pathway is predominantly involved in cell survival and cell growth. Activation of this pathway by mutations that confer constitutive activation of AKT result in only 10% of AMLs, but a vast majority of the mice come down with an MPN that quickly progresses to a T lymphoma. Another method of activating the PI3 kinase AKT pathway is by ablating expression of PTEN, which is a negative regulator of the PI3 kinase AKT pathway. Also, it's a tumor suppressor. So PTEN null mice, 3/4 of them come down with a T-ALL, whereas only 1/4 of them come down with a mixed T-ALL AML phenotype. So it seems that oncogenic RAS signaling predominantly gives rise to myeloid diseases. However, PI3 kinase AKT pathway predominantly gives rise to lymphoid diseases. So this seems to suggest that there's some other pathways must be mediating oncogenic RAS signaling. So as I mentioned, RAS can also activate the MAP kinase pathway. However, it's unclear exactly what kind of the most downstream portion of this pathway, MEK and ERK, can play, what role they can play in leukemiogenesis. So there's some data that has already been generated that shows that expression of active MEK in hematopoietic cell lines that are cytokine dependent, it can relieve the growth factor dependency. Also, expression of active MEK in 3G3 fibroblasts transforms those cells so that they're able to form foci on soft agar. Additionally, injection of these fibroblasts expressing active MEK in skin mice results in tumor formation. So while this data is very suggestive, it's all done in cell lines and primary or data in primary cells showing the role of MEK is lacking. So this is where essentially my project takes off. So there's two aims to my project. The first one being to determine whether active MEK signaling in primary cells can contribute to leukemiogenesis. And the second aim is to determine whether coupling active MEK expression with BCL2, another proto-oncogene, in HSCs, if that affects disease phenotype or disease progression in any way. So I'm going to start out by talking about aim one. And so to address the first aim, we used a retroviral transduction system. So we generated a retroviral vector that expresses a constitutively active form of MEK. In this retroviral vector, there is an iris GFP, so all productively infected cells should be GFP positive. So we use this retroviral vector to produce retrovirus. We then sort out HSCs, or hematopoietic stem cells, that we define as lineage negative, c-KIT high, STAR1 high. When we infect these cells, we are then able to sort out the infected population using GFP, and then we inject them into sulfate-related radiated break knockout mice. So when we examined the mice, this is the data that we generated here. So approximately 6 to 10 weeks post-injection, all the mice reconstituted with HSCs expressing active MEK died. And looking kind of more grossly, we find that the mice have splenomegaly, as you can see here. They also have increased cell numbers in the spleen. And consistent with Jalin's data, the skew in development occurs towards the myeloid lineage, and this is occurring at the expense of both B and T cell development. So we then wanted to look at bone marrow, which is the normal site of hematopoiesis, to determine whether hematopoiesis is affected, and indeed we find that it is. So looking at bone marrow aspirates, we find that in control mice, there's a variety of different cells, so lymphoid cells, myeloid cells, erythroid cells, and also in the active MET mice, we predominantly see myeloid cells. So looking at peripheral blood as an output of hematopoiesis, we find that it's quite dysfunctional. So there's a decrease in the red blood cell number, decrease in platelet numbers, and increase in leukocytes. And also kind of mirroring what we see in the spleen, we also find that there's a decrease in lymphocyte percentages and increase in both monocytes and neutrophils. So we also wanted to examine non-lymphoid organs to see if there was any type of infiltration in those organs, and indeed we find that there's myeloid cell infiltration in both the liver and the lung, and in vector mice we find that in the spleen there's a clear demarcation between red and white pulp. However, the splenic architecture in active MET mice is completely disrupted. So kind of all this data put together seems to suggest that active MET can result in a mixed MDS-MPN phenotype in recipient mice. So as I mentioned previously, MET-ARC signaling can affect numerous different cellular functions, one of which is cell survival. So we wanted to see if cell survival was in any way contributing to the disease phenotype that we're seeing.